So now I am gonna share my screen to all of you. Can you tell me, do you see the screen of my laptop? Okay. Do you now see the screen? Okay. So let's start with the presentation. The CRISPR-Cas9 gene editing from basics to applications. Explore the revolutionary gene editing technology that is transforming biology and medicine. So if you feel any problem in presentation and this screen that I share with you, you can ask me in in the chat box or by opening the mic. So let's start with the with this. So Albert Einstein saying is, if you can't explain it simply, you don't understand it well. Yes. Welcome to this International Molecular Biology workshop that is about CRISPR-Cas9 from basics to applications. Let's introduce myself. My name is Ali Tras, and I am a genetics and molecular biology scholar at University of Health Sciences, Lahore, Pakistan, brings a wealth of knowledge and hands-on experience in bioinformatics, stem cell therapy, genomics, genetics and evolution, and molecular diagnostics. With a keen interest in advancing scientific discovery, Ali is dedicated to inspiring others through engaging learning experiences and fostering a deep understanding of cutting-edge molecular biology techniques. So let's start with the CRISPR. The introduction of CRISPR. The full form of the CRISPR is cluster regularly interspaced short palindromic repeats. It means that there is a sequence, for example, and there is a sequence that is regularly clustered. That is regularly clustered interspaced short palindromic repeats. So there are parts where there are repeats like A, A, and so on, and that is G, G, and G. That are the palindromic repeats. That are palindromic repeats that can join, that are A, T, C like this, and C, T, A. That are actually the palindromic repeats. It is a core component of bacterial immune systems. Okay, CRISPR is actually a natural system of bacteria that are capable of cutting DNA, that cut the DNA and has been repurposed as a gene editing tool. CRISPR acts as a precise pair of molecular scissors. Okay, that is actually work as a molecular scissors that cuts the DNA. That cut a target DNA sequence directed by a customizable guide. This precision allows for highly specific genetic modifications. So, uh, now I'm going to tell you about how CRISPR works. So, CRISPR has actually this system, the CRISPR-Cas9, consists of two key components. Okay, first of all is the CRISPR-associated nuclease that is called Cas nuclease. Okay, that nuclease means that cut the sequence, that actually cut the nucleotide sequences. That is why it is called nuclease. It is actually an enzyme which binds and cuts DNA. Actually, that binds with it and cuts it. And a guide RNA sequence which directs the nuclease to its target. Actually, uh, there is a system that's like a guided RNA, okay, and this is a sequence that actually binds with the DNA. Okay, that actually binds with the DNA, and this sequence that is highly complementary to that DNA cuts the DNA. And this guided RNA is actually used to direct the Cas nuclease to its target. So the importance of the CRISPR is once its DNA cleaving mechanism was understood, CRISPR was rapidly developed as a genome editing tool. Okay, first of all, it is the natural process. Okay, that is understood by scientists. Then people know about it and thinks that it can also be used as a gene editing tool. Uh, Sorry, actually ignore my writing because with mouse my writing is very poor. Uh, that actually revolutionizing the genetic engineering. So the importance of CRISPR is precision and accessibility. CRISPR allows the scientists to rewrite the genetic code. Okay, that can rewrite the genetic code. It means that we can add genes, we can uh, cut the genes, we can knock out the genes by using the CRISPR in almost any organism. Why? Because it is a simple, it is a cheaper and more precise than the previous gene editing techniques. So the real world applications actually CRISPR because its potential spans from curing the genetic diseases that is present today in the world, like sickle cell anemia. Sickle cell anemia is actually a genetic disease, and it's not only a genetic disease, but it is also a single gene disease in which one gene is affected, and that due to that gene that is affected cause sickle cell anemia. Okay, and if we uh, we can make and check this gene, uh, either we activate that gene or inactivate that gene. So by this, the sickle cell anemia can be cured. And from genetic diseases to creating drought-resistant crops. Okay, from the man diseases to human diseases to creating the drought-resistant crops, addressing global challenges. Okay, that is the vast applications of CRISPR that we discussed in today's workshop. So the history and discovery of CRISPR. So CRISPR breakthrough was led by Dr. Jennifer Doudna and Dr. Emmanuelle Marie Jalpenici. Their groundbreaking paper in Science 2012 revealed that CRISPR-Cas9 bacterial immune system could be repurposed for gene editing. Okay, it's a Nobel. They got the Nobel Prize in 2020 in chemistry, the first all-female team to achieve this. So key contributions to CRISPR development, and one of them is Fan Zhang in 2013 pioneered the use of CRISPR in eukaryotic cells at the Broad Institute of MIT and Harvard and discovered novel gas variants. George Church in 2012 at Harvard Medical School, he was among the first to demonstrate the CRISPR's utility in human cells, paving the way for therapeutic application. Actually, this one is actually the key contributor in CRISPR development because they actually demonstrate how to use the CRISPR in human cells. And the last one is the Virginius Sixth, actually 2012, independently discovered the CRISPR gene editing capabilities on other organisms. Okay, that one is in humans and that is in other organisms, highlighting the parallel scientific breakthroughs. So the CRISPR mechanism, how does it work? So we dive into the molecular process that allows the CRISPR-Cas to precisely identify and modify the genetic sequences. So CRISPR system in microbes, adaptive immune system. Actually, it is the adaptive immune system in bacteria in the microbes. CRISPR system forms the basis of adaptive immunity in bacteria and archaea, protecting them from viral infections. When the virus attacks, when the virus attacks, that one is actually the virus, that one is actually the bacteria. When it attacks, they use the adaptive immunity and protecting themselves from that virus. So they use Cas nuclease. It uses Cas nuclease enzyme that bind and create a double-stranded breaks in the DNA. Actually, the viral infection or the virus inject the DNA into the bacteria. Okay, that is the bacteria and having a DNA of virus. So the Cas nuclease binds that DNA and cut it. And viral DNA snipping. When bacterium is infected by virus, it uses Cas nuclease to snip off a piece of viral DNA known as a protosphere. So this is actually a DNA. This snip of small amount of DNA, that one, that can actually be used as a memory for future infection. Okay, Mandeep, if you say voice is not clear, do you feel like this time, Mandeep, do you understand
सर सर
now?
आवाज कट कट के आ रही है।
Okay, do you hear right now clear or not?
सर अभी भी आवाज ना ऐसे कट कट के आती है आपकी।
Okay, I think so due to internet problem. It is okay. If you feel okay, I will also provide you the recording. That is that will be definitely be cleared. Okay. Sorry, it is the first time that my voice is not clear. I think so it is due to the internet problems because I am in Lahore and there is a flood in Lahore and there is a cable problems and my internet is actually a cable internet. So I think so the problem is due to this. Hope so you understand it. Thank you for understanding. So CRISPR systems in microbes immune memory. Okay. So the snipped viral fragment is stored in the bacterial genome alongside fragments from other viruses that have previously infected the cell, creating an immune memory. Okay. I will say here the DNA, the viral DNA, the bacteria snipped off small amount of DNA from it. Okay. And that can be used as a immune memory. The viral spacer fragments are precisely placed between the repeated palindromic sequences. This unique arrangement of spacer and palindromic repeats is what gives CRISPR its distinctive name. Okay? It is that spacer fragments that are actually present in the immune memory are precisely placed between the repeated palindromic sequences, and due to this, the CRISPR gets its name CRISPR. So CRISPR system in microbes targeted defense. Upon reinfection with the same virus, the bacterium can recognize and destroy it with highly specific Cas9 enzyme. Cas9 activity relies on CRISPR RNA. So CRISPR has a two components, CRISPR RNA and trans-activating CRISPR RNA. Okay. The CRISPR RNA is complementary to the virus spacer. Okay. This one is the virus spacer and the CRISPR RNA is completely complementary to the virus spacer. While the trans CRISPR RNA serves as a scaffold, okay, that actually contributes to the CRISPR RNA. These two RNAs, the CRISPR RNA and the trans-activating CRISPR RNA, can be used as a complex, and that actually known as a guided RNA, that essential for precise viral target. So this one is the case study, genetically modified mosquitoes. The first genetically modified mosquitoes developed by biotechnology firm Oxygen. This one is the actually the company that actually creates. It's a USA based company. Have been released. These are the male AIDS Egyptian mosquitoes designed to transfer a lethal gene to their female offspring, causing them to die before reaching adulthood. So by go through a proper cycle, so they die before reaching the adulthood. So, Aedes aegypti mosquitoes are notorious carriers of a devastating diseases like Zika, dengue, chikungunya, and yellow fever. This genetic approach aims to reduce wild mosquito populations and curb disease transmission. So, we now talk about the gene knockout and gene knock-in, and there are two techniques. One is non-homologous end joining, and another one is the homology-directed repair. So, CRISPR-Cas9 gene editing primarily function creating double-stranded breaks. Okay, so this one is the DNA of actually the viral DNA, and they cut by double-stranding break. Okay, the both of the DNA is break this time. The cells inherent DNA repair mechanisms. Okay, in the cell there is a inherent DNA repair are then strategically exploited can be used to achieve the desired genetic modifications. Okay, the first one is the non-homologous end joining, and the another one is the homology directed repair. In the homology directed repair, we need a template, and in non-homologous end joining, we do not need template. We do not need template. Non-homologous is error-prone repair. Is an error-prone repair. That is why it is used in gene knockout. That ligates that broken DNA ends without homologous template. Without there is no use of template. It often results in small insertions or deletions at the repair site. Okay, exploitation. What are the uses of a non-homologous antibody? Is deliberately used to disrupt and render genes non-functional. Okay, that actually non-functional the genes. Actually, gene knockout leading to gene knockout. So, in the homology directed repair, is highly fidelity repair pathway that uses a homologous DNA template to accurately repair the double-stranded break. This template can be provided exogenously, that are externally provided, that are externally provided. And the exploitation of the uses homology directed repair is the leverage to insert new genes or specific fragments of genetic material at the target site, achieving precise gene knock-ins, as in that is used to function the gene. Okay, and that actually render genes non-functional. So DNA repair pathway are two non-homologous and joining and homology directed in gene editing. So this one is the Cas9, and this one is the single guided RNA, and that actually do the double-stranded break. Okay, so there are two repair systems. One is the non-homologous end joining, and another one is the homology directed repair. Non-homologous repair pathway that leads to an insertion or deletion of nucleotides, potentially resulting in gene knockout. Okay, so remember it. If there is a non-homologous end joining, there is always a gene knockout. Okay, and if there is a homology directed repair (HDR), then always there is a gene knockout. Okay, for example, there is a non is a gene knocking. Okay, and there is a insertion or a deletion. So there is a knock out. So if you have any questions, you can ask me here. There is a section, there is a first section of question and answers. You can ask me questions here. If you have any questions, you can ask me.
Assalamu alaikum.
Okay, I think so. No one has a question, so we dive into the next part of the workshop. So I'm gonna share a screen to all of you. Okay.
Assalamu alaikum, Doctor.
Walaikum Salam.
What are the key differences between homologous and non-homologous?
The key differences between homologous and non-homologous.
Yes, you got.
Okay, in homologous, homologous
Hmm?
directed repair, we actually need a template. Okay, do you know about the template? Template DNA.
Come again?
Do you know about the template DNA?
The network is fluctuating. I'm not hearing perfectly.
Again, sorry.
I said the network is fluctuating. I'm not hearing perfectly.
Can you write your question in the chat box, in the message box?
Okay.
Because I can't hear you well.
I mean, my question is the difference between homologous and non-homologous that you have mentioned in your lectures.
Okay, okay. So in the homologous,
Mm-hmm.
we need a template DNA. Okay, so it's a
Okay.
template DNA that is used as a reference. Okay, if there is a
Mm-hmm.
double stranded break, then there will be a palindromic sequences of the homologous sequences that can be used to create or repair the DNA. That DNA that is actually affected. Okay, if we need template to repair
Mm-hmm.
that DNA, that is homologous directed repair. Okay, because why
Always.
homologous directed repair? Because
Mm-hmm.
we need template,
Mm-hmm.
and in non-homologous end joining, there is no need of template because we don't need donor template. So there is a error from. Okay.
Okay. Only the homologous needs a template, but non-homologous no need of template.
Yes. Yes.
Okay.
Exactly.
And
Exactly.
secondly, the homologous, and it is also used to create to the active.
Yes. 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 To active
Mm-hmm.
or to actually to fit to add the gene into the DNA. For example, there is a DNA of a mouse.
Mm-hmm.
Okay,
Okay. Yes.
and we need to add the gene into that mouse. Okay, just in order to
Yes.
check what actually that gene works. Okay,
Mm-hmm. Yes.
so for this we exploit, we use HDR, the homologous directed repair. Okay, in
Mm-hmm.
order to knock in means to enter, to fit.
Mm-hmm.
uh, so that is why we need homologous directed repair. But
Mm-hmm.
in non-homologous end joining, we don't need to add the gene, but we actually need to knock out the gene, to delete that gene, to delete the function of that gene. So for this, we need non-homologous and joining.
Mm.
Okay,
Yes, exactly understood.
thank you. If you
Mm.
have any
Thank you.
question, you can ask me.
Mm. Okay.
Okay, I'm gonna share the screen to all of you, and let's start the another um, part. Okay. Also, you all see the screen. So CRISPR gene knockout creating a non-functional genes. Uh, let's describe it. When Cas9 creates a double-stranded break in the DNA, the cell most common repair mechanism that responds is the non-homologous end. Okay, because in the double-stranded breaks, the mostly common repair is non-homologous end joining because there is a no template break. So it's an error-prone repair, as we all know. Non-homologous is a quick but imprecise repair pathway because it's an error-prone. It is frequently results the small insertions or deletions at the site of the break. So it's an error-prone repair, and it's also cause the frame shift mutations. When these indels, insertion and deletions, occurs within the coding region of a gene, they often cause a frame shift mutation altering the entire downstream protein sequence. Gene inactivation knockout. A frame shift typically leads to a premature stalk down or a completely abnormal protein. Okay? In non-homologous end joining, the gene knockout, we actually inactivate the gene. Okay? And how it is done? By frame shift mutations. In the frame shift mutations, we actually shift the frame. As we all know, there is the A, G, C, T, for example, and C, T, A, 2, 4, 6, 7, and T, for example, this. So it creates a triplet, AGC, TCT, and that ATC, for example, it's okay. So if there is a deletion of C, okay, there is a deletion of C. So what will be happen? AG, TC, TA, TC. Okay. So now the gene codon is ATC, AGT, CTA, and TC, and the next one maybe after TCT is the G. Okay. So the amino acids that be formed is different. Okay, and that is called frame shift. Hope so you understand. So this abnormal proteins formed by introducing the stop codons causes the gene inactivation and that actually rendering the gene non-functional. This deliberate inactivation is known as the gene knockout. So in the CRISPR gene knock in which we actually add the gene. When Cas9 induces a double-stranded break in DNA, cells can repair themselves via homology directed repair. This pathway allows the researchers to insert new DNA sequences or entire genes, a process known as gene knocking. To achieve a gene knock-in, a specific DNA template called donor template is required for the repair process. So CRISPR gene knocking the mechanism. First of all, we need to design the donor template, okay, by using in silico tools. So in the donor template contains the desired sequence of the gene. The, the gene or the sequence we need to make to in order to add into the DNA, flanked by a regions of homology. Okay, that precisely match the DNA surrounding the Cas9 cut site. And when we design the donor template, then we deliver it to the cells. The donor template is delivered into the target cell alongside the Cas9 enzyme and single guided RNA. And after it precision integration, these cells natural homologous directed repair machinery then uses the donor template. Use that donor template as a guide to accurately insert the new DNA sequence at the precise location of the double-stranded break. Okay, there is an we need exogenously externally we provide the donor template into the cell, and then the natural system of the homologous directed repair is used is uses the donor template as a guide to accurately insert the new DNA sequence at the precise location of the double-stranded break, where the double-stranded break actually happens. So gene knocking, there is abundance applications in biotechnology and human health. Gene knock-ins have revolutionized biotechnology, enabling advancements. For example, the production of recombinant proteins. We need to produce the recombinant proteins that are not naturally produced in that human or in that animal. Okay, but we need to produce that proteins. For example, for insulin. So we produce that recombinant proteins, insulins, in by using the CRISPR-Cas9 mechanism or by using that system, we use gene knock-ins. And it is also used to increase the viability of immortalized cell lines. Okay, if we used different types of cell lines that are immortalized, that are not died, okay, but we need to make it viable. Why viable? Because if cell lines are repeated again and again in the cycle, so there is a chance to introduce the errors in the cell lines. Okay, but we need to make it viable. In the viability, there is a chance, there is a probability to not to die. First of all, not to die. And the second one, there will be not introduction of errors. So this can be done, this can be increased by using gene knock-ins. And at the end, precision disease modeling. Okay, and the precision disease modeling is actually in which if there is a mutation in someone in someone a person, we can use as a model by using gene knock-ins. And crucially, CRISPR knock-ins holds immense promise for cell and gene therapies. Okay? In the cell and gene therapy, offering a pathway to correct gene mutations responsible for numerous human diseases. The precision allows for targeted interventions with fewer off-target effects. So in the gene knockout, we actually remove the gene, that one, and in the gene knock-in, we actually add the gene. And the third one is the CRISPR activation and CRISPR inactivation. Uh, in CRISPR activation and CRISPR inactivation, we don't need to break the double-stranded DNA. Okay. In this CRISPR activation and CRISPR inactivation, we actually regulate the gene expression. We actually increase or decrease the gene expression, not we add or remove the gene. In CRISPR activation, used to increase, upregulate the expression of it. Okay, the gene expression is actually producing the gene to RNA to protein. We actually increase that cycle. That is actually, and CRISPR interference or CRISPR inactivation used to reduce the downregulate the expression of a gene. And how they work? They use the dead Cas9. Okay, they use dead Cas9. Why? because both technologies, CRISPR activation and CRISPR interference, use that modified Cas9 enzyme called catalytic dead Cas9, Cas9, dead Cas9, which can't cut the DNA. Okay, there is no need to cut the DNA. Okay, there is just need to upregulate or downregulate or reduce or increase the gene expression. So transcription factors. That Cas9 is fused with transcription activators, CRISPR activation or repressors, CRISPR-I. Okay, there is just used transcription factor targeting the gene of interest. DCas9, the dead Cas9, the catalytically dead Cas9 complex is guided by a single guided RNA. There is also use of single guided RNA directly to the gene of interest, modulating its expression without altering the DNA sequence. That is actually called epigenetics. Okay. So these precision gene modulators have wide-ranging applications in various research fields, providing unprecedented control over gene activity. So CRISPR activation and CRISPR interference key applications. First of all, developmental biology. Okay, we can also create the mouses, the different types of organisms by using CRISPR activation and CRISPR interference. Because by this we can also control the gene expression, investigating gene roles, where the gene actually works in the cellular differentiation, and organism development. In the infection disease, we can use as a modeling, studying host pathogen interactions where host and pathogen interact, identifying the novel therapeutic targets, and the disease progression. We can use as a mapping the genetic pathways involved in the chronic diseases like cancer or neurodegeneration, functional genomics, and that is elucidating the function of uncharacterized gene across the genome. Drug resistance screening, identifying the genetic elements that mediate resistance to various therapeutics. So, if you have any questions and answers, if you have any questions, you can ask me here. There's a question and answer section. If you have any questions, you can ask me here. Mhm. Okay, listen, everyone. After 5 minutes, this meeting will be end. So we have to join the new meeting. So I am gonna share the meeting link in the chat box. Kindly join that meeting. Hope so you understand. I'm gonna share the joining link. Kindly join that link. We're gonna left this meeting in 5 minutes. Kindly join that link, and we continue the next part of the workshop in silico and the applications and the future prospects of the uh, CRISPR-Cas9 in the next meeting. Kindly join that meeting. Kindly everyone join that meeting and left this one. Tahreem and Musa Abdullahi kindly left this meeting and join the new meeting by using the link.
Okay.
Tahreem also you also kindly left this meeting. Tahreem, do you hear my voice? Tahreem, do you hear my voice? Kindly left this meeting and join the new one. Nuwan Ismail and Tahreem kindly join the new link. Everyone kindly join the new link. So I think so everyone is here, and we're gonna continue the workshop. So I am gonna share the screen to you. Hold on. Can you tell me I am hearable? I am audible?
Yes, sir.
Okay. And screen is showing to all of you. Okay. So let's dive into the application of CRISPR technology. So first of all, the cell and gene therapies, revolutionizing the medicine. The CRISPR holds immense promise for curing the wide range of genetic diseases, okay, including the neurodegenerative conditions, cancers, blood disorders. The first clinical trial of the CRISPR cell therapy initiated in 2019 focused on treating patients with sickle cell diseases. The groundbreaking treatment successfully restored the fetal hemoglobin production, that are produced in the fetus, eliminating the need of a functional adult hemoglobin copy. Okay, the adult hemoglobin copy is not functional in that patient. Okay, so they restored the fetal hemoglobin production. So by this, they eliminate the need for functional adult hemoglobin copy. So cell and gene therapies. The next one is the CRISPR enhanced immunotherapy. CRISPR technology is being harnessed to generate human antigen receptor that is called CAR T cells. Okay, the CAR T cells is actually attack the cancer cells. Okay, a powerful form of immunotherapy for cancer treatment. Okay, for cancer. This process involves extracting the T cells. They first of all extract the T cells from a patient's blood. Patient's blood, they extract the T cells, genetically modified their DNA. They genetically modified their DNA to express the CAR, the CAR, the chimeric antigen receptor, and reinfusing them back into the patient. Okay. First of all, they extract the T cells and then modify. Okay, and then reinfuse back into the and then reinfuse back into the patient. This engineered CAR on these T cells enables them to specifically recognize that they actually increase the specificity and bind to the target antigens on the cancer cells, triggering the T cells to activate and destroy the malignant cells more efficiently. So future horizons broadening the scope of genetic cures with the still early stages. There is a very still a early stages. CRISPR technology is poised to treat thousands of genetic conditions in the future. Potential applications include targeting the breast and ovarian cancers linked to BRCA mutations. Okay, they actually cure the BRCA mutations. Huntington diseases, Tay-Sachs, beta thalassemia, cystic fibrosis, early onset of Alzheimer. This represents a paradigm shift. There is a mean paradigm shift is means the shift in the treatment of the inherited disease. So the main one are the recommendations for the ovarian and breast cancers, Huntington disease. There is the neurodegenerative disease, early onset of Alzheimer's, and the cystic fibrosis. The second one is the diagnostics. Okay, so CRISPR-Cas9 is also used for diagnostics. CRISPR in disease detection. During the global COVID-19 pandemic, CRISPR emerged not only as a potential therapeutic tool but also a crucial diagnostic instrument for coronavirus. So this one, like the Sherlock CRISPR SARS COVID-19 test, it a rapid, early, and highly accurate diagnostic method. So this company that produces Sherlock CRISPR SARS COVID took a test kit that actually exploit CRISPR-Cas9. So they got emergency use authorization from US federal authorities for use in the laboratory settings. This marks significant milestones in the pandemic response. So advanced CRISPR-based COVID-19 diagnostics, like we use the detector that actually detect or the diagnose, and CRISPR-based methods like Sherlock, stop COVID, and that leverages the highly specific search function of the Cas9 and other naturally occurring Cas9 nucleases, for example, Cas12 and Cas13. They don't only use Cas9, but the other types like Cas12 and Cas13 to accurately detect the viral genetic material. This mechanism allows for the rapid and precise identification of the pathogen. So third one is agriculture, cultivating a sustainable future. Gene editing technology, especially CRISPR, holds immense potential for transforming agriculture with its first, predicting CRISPR-modified foods will be widely available within 5 to 10 years. Its primary application lies in the creating crops that are inherently disease resistant. Okay, they produce the crops that inherently reduce disease resistance. In their inherent, there is a disease resistance and the drought resistance. So if the water is low, they don't be that, enhancing the global food security. So, for example, scientists produce cacao like the chocolate plants using this technology. So they also extend the shelf life of various foods, reducing waste. So in Pakistan, the Center of Excellence in Molecular Biology is actively working on CRISPR engineered varieties resistant to drought, salt, verticillium wilt, and viruses and heat. So in the bioenergy, fueling the innovation, actually in bioenergy, we actually increase the algae. We actually increase the viability of the algae. So, significant advancement in the bioenergy. Scientists have successfully utilized CRISPR to knock out multiple transcription factors that regulate the lipid production in the algae. Actually, they knock out the regulations of that lipid production. So, actually, they regulate that transcription factors that actually down regulate the lipid production. But we actually increase it. So, we knock out that transcription factors. This precise genetic modification has led to the